Now let's pick back up with translation. So we have an mRNA copy of DNA. Remember, mRNA is single-stranded, it's easy peasy. It's basically a disposable copy of that original instructions in DNA. Yeah? So at this point in time, we have an mRNA molecule already constructed, already gone through transcription. That mRNA is now going to pop out of the nucleus, head out into the rough ER, maybe find a ribosome there, maybe it'll find a ribosome in the cytoplasm, but the mRNA is going to connect up with the ribosome. So let's see how this plays out. We're going to start first with talking about the key players here in transcription. So we've touched on these before. You may not remember them. Hopefully you do. But the key players in translation, we have the ribosomes, right? That's the organelle that builds proteins, synthesizes the polypeptide chain. So really what it's doing, the ribosome, it's kind of acting like an enzyme in the sense that it's going to catalyze the bond between amino acids, between the carboxyl end of one amino acid and the amino end of another. Remember, it's going to build that peptide bond. You should be able to identify what that peptide bond looks like on a molecule. tRNA. tRNA is another type of RNA. It is called transfer RNA. It's what's carrying the amino acids to the ribosome. Ribosome doesn't know which amino acids to connect together. It just knows how to connect amino acids. tRNA basically is what helps the ribosome connect the right amino acids in the right location of this protein. tRNA has a region on it that is very important. It has a region that we call an anticodon. So these three pieces right here, those are bases. Those could be an A, this could be a U, this could be a G, this could be a C, but they're three bases that will be complementary to the three bases of RNA. So RNA is read in triplets. Every three bases of RNA results in one amino acid. So these three anticodons on the tRNA are complementary to the three bases on the mRNA that we call a codon. So they match up. And that's how the ribosome knows which amino acid to connect together in which location. As long as this anticodon matches up with that codon, good to go. So again, that codon's that three-letter sequence of RNA that will match up and be complementary to that three-letter sequence on the tRNA. Okay, ribosome, tRNA, mRNA. tRNA is carrying the amino acids. Ribosome connects together the amino acids, builds that polypeptide bond. mRNA just contains the instructions. It contains the codons for which amino acids go where. So we've actually cracked this code of the codons. We read the genetic code in a triplet sequence, which again we call a codon. So every three bases is a codon that corresponds to one amino acid. So here's how we read this. So we have the first letter in our codon is up here on this left-hand side. The second letter in our codon is up here along the top. And the third letter in our codon is down here along the right-hand side. So let's say we have the we have the nucleotide sequence of GCA. Let's say GCA is our nucleotide sequence. GCA. So here's G. Go along here till we find C. And A is right there, right? So those three nucleotides in mRNA, GCA, tells that at that location in that protein, we should have this amino acid. This ALA stands for alanine. So the alanine amino acid should go here at that location if we have a GCA codon. So that's how DNA is read, basically, or ends up finally in protein, is these codons that are containing in the mRNA will be used to match up with the anticodons of the tRNA, and that's how we connect together the amino acids. All right, translation step 1. First thing that happens, our ribosome kind of looks like a hamburger buns, you could say. So we have two subunits to our ribosome. We have the big top bun and the smaller bottom bun. So first thing that happens is this lower bun, the smaller subunit of the ribosome, is going to connect to the RNA molecule. There's going to be tRNA that comes in that has an anticodon that matches up with the very first codon of the mRNA. All proteins have their very first codon, their very first amino acid codon in there is AUG. AUG is what we call the start codon. So if we go back to our chart, AUG right here, it's in green because that is our start codon. So what starts off all polypeptide chains is an AUG, which is the amino acid methionine. So we have small subunit, the mRNA binds to that smaller subunit, and now we have a tRNA that has a complementary anticodon to that AUG start codon, and it's carrying the amino acid methionine. Okay, next, the larger subunit, the top bun, comes and sits on top of it all. The top bun is really what's doing the catalyzation of binding those amino acids together. So we have, here's our first amino acid right here, methionine, being carried by this tRNA right here. It is sitting in what we call the P site, okay? And it's connected to the AUG anticodon. Remember, A and U bond together, A and U bond together, C and G bond together, so it's complementary. Next, our next codon, UUU. Those three letters match up with AAA. So tRNA that has that anticodon will only be carrying the amino acid phenylalanine, right? Okay, so each anticodon corresponds to just one amino acid. And this is how we try not to mess up the protein structures, because each tRNA is specific to the amino acid it carries. All right, cool. So got the larger subunit come in. We have our next tRNA coming in. It's going to sit in what we call the A site. And basically what's going to happen, this ribosome, it has an active site, just like an enzyme. It's going to take those two amino acids and smush them together to make that protein bond. So whatever's in the P and the A site get connected together. We build that peptide bond by the ribosome catalyzing that reaction between the amino group and the carboxyl group of the different amino acids. And it's going to transfer, basically what happens is it transfers the amino acids, or the amino acid chain that's in the P site, it transfers them to this next tRNA that's sitting in the A site. And then it shifts over by 3. So here we have now, we've catalyzed methionine and phenylalanine. We've catalyzed that peptide bond. Now we shift over by 3 bases. We expose our next codon, CGA. The anticodon that's complementary to that, GCU, comes in. It's now going to sit in the A site. Arginine is what's being carried by this tRNA. Now we have phenylalanine, methionine, P and A sites right here. The ribosome is going to catalyze the reaction between this phenylalanine and this arginine and basically send this chain over, this growing polypeptide chain over to that location right here, added on to this arginine. So it's going to grab this phenylalanine methionine chain, connect to the arginine, and now we have 3 polypeptides. And then it's going to do it again. It's going to move over by 3 bases, expose those 3 bases. This one flies out. We get a new one come in, transfer that chain to the next one, and then continue. So it's going to continue this process of connecting the chain, moving over 3 bases, new tRNA comes in, connect the chain, moving over 3 bases, new tRNA comes in, until we hit a region that's called a stop codon. So a stop codon, we have 3 codons that do not correspond to amino acids. There are no amino acids that are connected with these codons. UUA, UAG, and UGA are these stop codons. So there are tRNAs that can bond to these locations, to these stop codons, but these tRNAs that connect to the stop codons do not contain an amino acid. There's no amino acid contained with or connected to those tRNAs. Okay? So it keeps going. Just keep chugging along. Just keep chugging along. Could be 200 amino acids, could be 500 amino acids, could be 10 amino acids. RNA is just going to contain the template for building that protein. tRNA contains a complementary base connected to amino acid. The ribosome catalyzes the bond between those amino acids. Eventually, we'll get a polypeptide chain. That polypeptide chain will be folded, and we'll have pieces added to it, maybe needs a cofactor or a coenzyme. It'll be altered or edited in the Golgi if it needs to, and then gets sent out to wherever it needs to go, right? It's constructed here, that polypeptide chain is constructed here at the ribosome. Any additional edits or folding, all that special stuff, that's all done by other portions of the cell, okay, in other areas. Ribosome just connects together the amino acids to make the polypeptide. Okay, so that sequence of DNA that we transcribed in the last video, let's now translate it into a protein. So I'm going to go back to my tablet and we'll translate that sequence of DNA that we transcribed earlier into a protein. Okay, so remember, DNA here is in purple, RNA here is in orange. Let's now translate this protein using our codon chart. Okay, so this isn't perfect because we didn't start with a start codon. Remember, if we had, it'd be AUG is our start codon. But that was my bad. That's all good. We can still practice though. Okay, so remember, DNA is read in triplets that we call codons. We got an extra one there. So using our chart, right? Left-hand side, we have our first letter of our codon. Top side, we have our second letter of our codon. And down the right, we have our third letter. So we use our codon. U is our first letter. Go along that row until we find A. And then go along that row until we find G. Whoops, that's a stop codon actually. That wouldn't work. But let's just keep going just for practice purposes. So now we have CUU. CUU gives us the amino acid leucine. ACC, go along that row of A till we get to CC, gives us the amino acid threonine. I'm not expecting that you know what these three-letter codes are. I just want you to know that they're different amino acids, right? And that they just represent different side chains to that amino acid. AGG gives us the amino acid AGG, go along the row of A, we hit G, arginine. Pirate's amino acid, arginine. And GUG gives us the amino acid G to U to G valine. Okay, remember, this was my bad. I started off with a stop codon accidentally. Usually that stop codon would be at the end over here. So typically we'd have a stop codon over here. So we would typically have something like UAA here, which would be a stop. We also got another base in here. Let's pretend this is an exon, and this is an intron. We're going to take that intron out. Okay, so again, ignore this, my bad. But we have leucine, will be bound to threonine, bound to arginine, bound to valine, and that's our sequence, that's our polypeptide sequence. Okay, we could keep going. Usually in most proteins there are thousands of amino acids, but we'll just stop here for example's sake. So first, second, third base, find that combination on our codon chart, and that tells you the sequence of amino acids that go in that protein. And now we've just completed translation. Yay!